Having a good liver function is very essential to survive because the liver is a very vital and important organ that is responsible for many metabolic and synthetic processes. So that it is very important to have an equal liver function to survive again. Okay? Nobody can survive more than 24 to 48 hours if he has no liver. Person in anhepatic state can't survive more than 48 hours, and this is pretty good to know. Okay, this indicates how important is the liver. Let's move to that table. This table summarizes the main functions of the liver. Okay, it may be hard to recall all these points, but it is pretty easy to recall it if you just organize them in a good way. I suggest to make two groups of functions of the liver. The first one, metabolic function. Okay, just metabolic. This color is not good, not very clear. Let's change it to red. Okay, much better. Metabolic function. And the second group is the synthetic function. Let's start with the first point here, maintaining core body temperature. I won't get into details of maintaining core body temperature function of the liver, but I can say that it is the result of some metabolic functions of the liver. So I will add it to the first group, the metabolic group. Okay. The second function of the liver is pH balance and correction of lactic acidosis. And again, this is a metabolic function of the liver to balance pH. I will get over the third function because it is synthetic, and I want to talk about the metabolic functions first and then move to the synthetic. Glucose metabolism, glycolysis and gluconeogenesis. Okay, these two functions, glycolysis and gluconeogenesis, is a very important function of the liver, and it is clear that these are metabolic functions. The glucose metabolism is metabolic function of the liver. So until now we have temperature, pH balance, glucose metabolism, and the fourth point is urea formation from protein catabolism. Some metabolic pathways or some metabolic processes in the liver lead to the formation of urea. And this is metabolic function of the liver. I will get over bilirubin and move to drug and hormone metabolism. One of the most important functions of the liver is to metabolize a lot of drugs, a lot of hormones. Examples of these hormones are estrogen and testosterone. These are two important sexual hormones that is metabolized in the liver. And if something bad happens to the liver, this will lead to some sexual abnormalities due to the abnormalities in metabolizing these two hormones. Okay, and the final okay, just and what is synthetic here, And the final metabolic function of the liver is dealing with gut endotoxins and foreign antigens. Dealing with gut endotoxins and foreign antigens. I believe that gram-positive cocci or cocci, whatever it was, is metabolized or removed in the liver after being derived into the liver by a portal vein. Okay, so removal of gut endotoxins and foreign antigens is an important metabolic function of the liver. So you may think it is hard to recall these, but it is easy. Temperature, pH balance, glucose, urea, drug and hormone. Glucose, urea, drug and hormone, and endotoxin dealing with is the metabolic functions of the liver. Just remember glucose, urea, bilirubin, glucose, urea, pH, temperature, and endotoxins, drug and hormones. It is very easy. Now let's move to the synthetic functions of the liver. The liver synthesizes some important things like albumin. I think it is not mentioned in the table, so that I will add albumin. The albumin is a carrier protein that is very important and essential in the serum. It is actually the most important protein in the serum, the albumin. Okay, this is the first thing synthesized in the liver. The second is the clotting factors. Almost all of clotting factors are synthesized in the liver. So albumin, clotting factors, and the third thing is bilirubin formation. It is not accurate to say bilirubin formation because it is not formation of bilirubin, is a conjugation. Okay, I will talk about it in a few minutes. Okay, so bilirubin, albumin, clotting factors. Bilirubin, albumin, clotting factors. These are three synthetic functions of the liver. The metabolic functions, as we said, temperature, pH balance, urea formation, temperature, pH, urea, glucose, drug and hormone like estrogen and progesterone, and endotoxin. Okay, let's move to bilirubin metabolism. The bilirubin is the result of red cell destruction or hemiprotein catabolism or bone marrow erythropoiesis. Okay, these three things can result in a bilirubin. Okay, bilirubin. Bilirubin in a blood attached to albumin. If you remember, we've just said that albumin is a carrier protein synthesized by the liver and circulate in the blood. In the blood, bilirubin attached to albumin. Bilirubin and albumin is called unconjugated bilirubin. Unconjugated bilirubin. Okay, this unconjugated bilirubin will move to the liver. In the liver, a process we call conjugation happens. Conjugation. So, the bilirubin is not unconjugated anymore. It is conjugated bilirubin now. After conjugation, the conjugated bilirubin moves to the biliary tree. And from the biliary tree to the intestine, and eventually get out of the body with feces. Okay, Some of this conjugated bilirubin does not get out with feces. It just goes with blood again and is excreted by the kidney. Small amount of conjugated bilirubin goes into this pathway. Okay, so if we have abnormal range of bilirubin in the blood, what is the possible causes, or what are the possible causes? The first possible cause is increasing in red cell destruction. Increasing in red cell destruction. As we said, that red cell destruction is the process responsible for bilirubin formation. So increase in red cell destruction is the first possible cause of increasing bilirubin. Okay, let's think about another cause. If the liver has something growing in it, and there is hepatocellular dysfunction, the liver can't excrete the bilirubin anymore, and the inability of excretion bilirubin will lead to increasing bilirubin in the body. So a damage in the hepatic cells is the second possible cause of increasing bilirubin. Okay, it is the second cause. The third cause, if we have normal red cell destruction, if we have normal hepatic cell function, but there is an obstruction in the biliary tree, here for example, here wherever it is, if we have obstruction, the conjugated bilirubin will not be excreted anymore, so that it will be increased, and this is the third possible cause of increasing bilirubin in the blood. Okay, so if we have abnormal bilirubin range, we have to think of one of these three causes. Okay, we have to know the normal range of bilirubin in the blood. It is between 5 and 17 millimole per liter. Between 5 and 17 millimole per liter. Sorry. Okay. This is the first liver function test. What other tests can we do? From the functions of the liver, we can know that the liver, as we said, do a metabolic processes, a lot of metabolic processes, or does a lot of metabolic processes. These metabolic processes need enzymes. Okay? Need enzymes. Okay. If there is anything wrong with liver cells, there will be something wrong with the metabolism. Okay? And the wrong with the metabolism will lead to abnormal enzymes level in the liver, or abnormal enzymes levels in the body. So we have to know the most important enzymes in the liver that we can use to know how is the liver. Is it good or is there are there some bad things happening to the liver. Okay, these enzymes are multiple. Four of them are important to us. The first one is alkaline phosphatase. Alkaline phosphatase, Al alkaline and P phosphatase. The second one is aspartate transaminase. Aspartate transaminase, AS is for aspartate and T for transaminase. The third one is alanine transaminase. Alanine transaminase. The fourth one is gamma glutamate transpeptidase. Gamma glutamate transpeptidase. Let's talk about them one by one. The first one is alkaline phosphatase. By the way, alkaline phosphatase is not exclusive for the liver. We can find alkaline phosphatase in many tissues, or in we can find it anywhere in the body. Okay, but it is significant in the liver. So, from the name alkaline phosphatase, phosphatase means that this enzyme do remove or replace a phosphate group in some complex. Okay, and we can see that here is the R is the phosphate group or phosphate group, and it is removed from the complex or replaced by an oxygen molecule in more specific words. Okay, alkaline phosphatase, it is what we call hydrolase enzyme. Hydrolase enzyme. That means that it uses a water molecule, okay, to replace it with a phosphate molecule. Okay, and that's what happens. Okay, it is not important to understand the reaction in details, but the important thing to know that alkaline phosphatase is one of the most important enzymes in the liver, that increasing in indicates a cholestatic or obstruction thing in the liver. Okay, obstruction or cholestatic wrong in the liver, deterioration. Okay, let's move to the second enzyme we've mentioned, the aspartate transaminase. Again, from the name, aspartate means we have aspartate molecule. Transaminase means that we have an amino group, and this amino group will be transferred to another complex. As we see here, the amino group is transferred from the aspartate to alpha-ketoglutarate to make glutamate and oxaloacetate. Glutamate and oxaloacetate. Very easy. Here, the amino group, here it is, and we form glutamate from aspartate and oxaloacetate from alpha-ketoglutarate. The important thing to recall that aspartate transaminase is not exclusive to the liver. We can find it in bone, we can find it in heart, we can find it in kidney and in musculoskeletal system. So elevation of aspartate transaminase is not always accompanied with liver dysfunction. Could be heart attack, for example, myocardial infarction, or something wrong with bone, with kidney, with musculoskeletal system. Okay, it is not that specific test. The third enzyme we use is the alanine transaminase. Alanine transaminase. From the name, we can know that its function is very near to the function of aspartate transaminase with changing the amino acid. Okay, the important thing to recall here is that alanine transaminase is much more specific than aspartate transaminase. That means that if we have increasing in alanine transferase ranges, that because the cause of this increasement in alanine transferase is more related to the liver than any other organ, though it can be due to other causes, but it is more specific. It is not 100 specific, but it is more specific than aspartate transaminase. It can be also due to kidney, heart, and bone, whatever, but it is more specific than aspartate transaminase. The fourth enzyme we use is glutamate gamma glutamyl transpeptidase. The important thing to recall that gamma glutamyl transpeptidase is an enzyme that is important in a case of acute alcohol ingestion. So, about each one of these enzymes, we have to recall something important. About gamma glutamyl transpeptidase, it is specific for acute alcohol ingestion. About alanine transaminase, the important thing to recall that it is more specific, okay? More specific than what? More specific than aspartate transaminase. Aspartate transaminase, we have to remember that it could be due to heart attack, osteoblast of the bone, and some kidney problem, whatever, okay? And the alanine alkaline phosphatase, we have to recall that it is not 100 specific to the liver, okay? This about the enzymes of metabolism of the liver, okay? We've said that the liver is a metabolic and synthetic organ. We talked about the metabolism. Now we will talk about the synthesis. Okay, we said that the liver synthesizes clotting factor, and we can investigate that by what we call prothrombin time. Okay, prothrombin time is for clotting factors. The second thing is the albumin, the important protein that is synthesized in the liver, the albumin. Okay, we see the range of albumin and we make sure it is normal range or not. If it is not, it could be the liver. Okay, this is all about liver function test. The final or the not the final, but there is still 2 points. The first one, you have to recall the normal range of these test or these substances in a good way, because it's pretty quite important. Okay, and the final point to say is that in chronic liver disease, if we have a patient with a chronic liver disease, the most important tests we have to do to monitor this patient is albumin, bilirubin, and prothrombin time. Albumin, bilirubin, and prothrombin time. Albumin, bilirubin, and prothrombin time. The normal range of albumin is between 35 and 50. 35 and 50. Bilirubin is 5 to 17. Prothrombin time 12 to 16. 12 to 16. So albumin 35 to 50, bilirubin 5 to 17, prothrombin time 12 to 16. Okay, this is all about the liver function and liver function test. See you in the next video.